Alright, hello everyone. Today we're going to be talking about the fluid mosaic model. The cell membrane is not a rigid barrier. It is a dynamic, selectively permeable structure. So here's how it is built and what each component does. So if we start with the phospholipid bilayer. So the foundation of the cell membrane is the phospholipid bilayer. So that is referring to this yellow section here. So each phospholipid, so if we look at an individual phospholipid, they have this general shape here. So each phospholipid has a hydrophilic phosphate head, which is attracted to water. So this one is attracted to water. And then we also have a hydrophobic fatty acid tail. So those tails are hydrophobic. So a better way to say it is we have a hydrophilic, which means it likes water, hydrophilic head, and then hydrophobic fatty acid tails. And we can see that if we zoom in, we have the hydrophilic heads, they're facing outwards. That just means that they like water. So remember, we have water on either side, so water here and water here. So the hydrophilic heads are going to be facing towards the water because they like it. And then these tails are going to be facing inwards away from the water. So these parts here are facing away from the water. Okay. So now let's look at these phospholipids, they arrange into that bilayer structure. And this arrangement, it forms a stable yet flexible barrier. So the fluid component of the model refers to the ability of these phospholipids to move laterally within their layer, giving the membrane flexibility. So these phospholipids will move laterally. So they'll move sideways like this, which gives the membrane its flexibility. Okay, now let's look at cholesterol. So cholesterol molecules, we can see on the diagram, are these green things here. So what they do is they're interspersed throughout the phospholipid bilayer. They regulate that membrane fluidity. So prevent the membrane from becoming too rigid at low temperatures and too fluid at high temperatures. So remember that cholesterol stabilizes membrane fluidity across temperature changes. And now let's have a look at proteins. So proteins are embedded throughout. So these are these blue things here. There's a few different types. So in general, proteins are embedded throughout the bilayer, the mosaic component of the model. They serve a range of functions. So integral proteins, which is this longer cylindrical shape here, they span the entire bilayer. Those that form pores or channels, protein channels, they allow specific ions and polar molecules to pass through the membrane that could not otherwise cross the hydrophobic core. Okay, and then we also have peripheral proteins here, which are these ones. They're attached to the surface of the membrane and are involved in structural support and cell signaling. We'll talk about that more when we look at active and passive transport. So there's also something else called glycoproteins. So if we look at glycoprotein here, so what they do, they're proteins with carbohydrate chains attached. They're located on the outer surface of the membrane. They function in cell recognition and communication, acting as identification markers that allow cells to distinguish self from no self. They're also involved in receptor functions, binding to hormones and other signaling molecules. Alright, so to summarize, the fluid mosaic model describes the cell membrane as a flexible phospholipid bilayer with proteins and other molecules embedded throughout. Phospholipids form the bilayer through hydrophilic and hydrophobic interactions. Cholesterol stabilizes fluidity. Protein channels facilitate selective transport of molecules across the membrane. Glycoproteins on the outer surface are involved in cell recognition and signaling. All right,